This is actually part 2 of our introduction to statistics. The last time we talked about univariate statistics and discussed some of the issues with respect to distributions, t-tests, u-tests, as well as the importance of p-values and the alpha, which is your cutoff, and then also issues about false discovery rates and multiple comparisons. So this is part of our last day, which is highlighted in blue-colored lecture titles. So we'll wrap up statistics, and then we'll dive into metaboanalyst, and we'll break that over 2 parts, and then close off with just sort of a blue sky view of the future of metabolomics. So we aren't going to be talking a lot about NMR. In some respects, we've done that for the last 2 days. This is basically how to deal with your NMR data and how to interpret and understand what you're doing. And we're using, and we'll be using Metaboanalyst as an example. It's not the only resource to do that, but we'll use that as an example. So let's start with statistics, and we'll talk a little bit about data comparisons. And this is, again, just sort of scenarios that we're looking at. Many types of experiments, whether they're medical or biomedical or agricultural or purely basic science studies. There's often a case where you're looking at a population before and after. So that might be time variations or before and after a treatment. Or again, it could be 2 situations with 2 different populations. But either way, we're trying to compare the dependency of one variable against another. In some cases, if we're working on prediction or modeling, which is also done in metabolomics and other omics sciences, we might be wanting to see how our observed properties, our observed metabolite levels, for instance, might match a predicted metabolite level from a model. And that could be applied to genes or proteins. So what we're doing, as with everything in statistics, is we're looking over a population of subjects and we're measuring multiple samples. So when we do these sorts of comparisons, we typically create a scatter plot. So this is a correlation between husbands and wives and their ages. And what you can observe from this plot is that generally most people marry similarly aged individuals. There's obviously some scattered to this, where there might be some individuals marrying younger spouses or vice versa. But this is just showing a trend, and it's not perfect. And it's not expected to be perfect. So what you're really showing is, or trying to identify with this sort of scatter plot, is to identify a pattern, a relationship, or what we usually call a correlation. So most of you are familiar with correlations, and again, this is just sort of a quick introduction to it, but it is something that's important because we've talked about covariance and correlation in our statistical spectroscopy lecture, and we'll see this in a few other occasions when we talk about metaboanalyst and looking at metabolomic data. So most people plot correlations as a positive sloped diagram. It is also possible to display a negative correlation. And then what's shown in the middle is essentially uncorrelated data. Now what you're trying to do typically is either qualitatively or quantitatively assess that clustering or those trend lines with a scatter plot. So qualitatively, we can identify something where there's a tight scatter and a clear trend, which is on your left, is a high correlation. Something where there is more scatter, a weaker trend, a broader cross-section to the data, if you want, that's characterized by a low correlation. And then if you have things on a perfect line, you have perfect correlation, which is never seen in chemical, physical, or biological measurements. So to quantify this, a fellow named Pearson came up with a formula to describe what's called the correlation coefficient. So instead of just simply saying correlation is high, medium, or low, or perfect, we can calculate this, which is really a measure of the scatter of these points. And so that correlation, which we called good or high, has a correlation coefficient of 0.85. The one that's not so good, a correlation of 0.4. The one that's perfect, a correlation of 1. So this gives a quantitation to something that we would normally just describe. And this goes back to my point about what statistics is. It's sort of the mathematics of intuition. So this is giving a number, and now people can talk in the same language and compare things. So the correlation is called the linear correlation coefficient alone, Pearson product moment correlation coefficient. And it is the standard way that many people use to assess comparisons and dependencies as well as predictions. Typically the predictions that might be arising from modeling or simulations or other kinds of computer tools or equations that you may have developed. Now there's, I guess, a pet peeve that I have about a correlation coefficient and something called the coefficient of determination. R is the correlation coefficient. R squared is formally called the coefficient of determination. R squared obviously is never negative. It is also the standard thing that's presented by Microsoft Excel. And I think this is a programming bug in Microsoft Excel because, in fact, you're supposed to really plot or display R and not R squared. Many people call R squared the correlation coefficient. It is not. It is the coefficient of determination. So I'm trying, although it's not succeeding, to get people to use R instead of R squared when they're talking about it, because this is the value that actually Pearson wanted people to use, and not R squared, and not what Excel tells people to use, or Microsoft. So we talked about significance and assessing statistical significance. So on the left is a plot where we can see a correlation, which is pretty good, coefficient of 0.85. There's perhaps 100 points there. And we can see another example where there's 3 points. And what you're seeing is a correlation with this of 0.99. So the question is the plot with the correlation coefficient of 0.85, is that value significant? And is the one with a correlation of 0.99, is that significant? And this is something that isn't too often or too frequently addressed or looked at. There's lots of tricks that people can play with generating really great correlation coefficients. And this is essentially the problem that you see, is that if we started measuring a few more points from 3 to now 5, we went from 0.99 to 0.05. So in this regard, obviously, it makes sense to have more data points to assess whether there is a strong relationship. As I say, there's ways of tricking the correlation coefficient. So you can get great correlation coefficients but are technically meaningless if you use only the data from the extreme ends of a curve. And this is done more often than you would think, where people might get on one end a modest correlation coefficient, and on the other end another modest correlation coefficient, but they put the two together with nothing in between, and therefore you get a fantastic correlation coefficient. And so this is something like having just the equivalent of 2 points in your plot. What should be done, ideally, is to treat each of the two clusters and assess the correlation coefficient on each of those clusters, or find data that fills out the middle. And again, the other trick that many people use is they just use a very small number of points, and then they can quote the best or optimal correlation coefficient, or they toss out the ones they don't like. And again, that's done surprisingly often. So to assess significance, it is actually possible to use a form of a t-test to determine whether your correlation is significant, determine whether the slope of the regression line is statistically different than 0, which is your null hypothesis. And as you might expect with any statistical thing, the more points, the greater the confidence in that correlation, or whether that slope is other than 0. So that's a brief discussion of correlations and I guess an appeal for people that if they're trying to generate correlations, either via their predictions or comparisons between two related groups, that you not only quote the correlation coefficient and not the coefficient of determination, but that you also quote the significance of that by providing some p-value. Now, because often in things like metabolomics, you can do things like modeling. You can predict certain things. You might have, again, an idea or a basis perhaps on a previous model or a previous model organism that you could sort of predict things. Predictions are pretty common in the field of cheminformatics and bioinformatics. We predict structure in biology. We predict performance and height and weight for different models, different ways of doing that. Now, in continuous variables, the best way of assessing those types of predictions is using the correlation coefficient. But with binary predictions, instead of yes, no, sick, or healthy, we use a different way of assessing those types of predictions. So in those ones, we use sensitivity and specificity, and we use terms like false positives, false negatives, true positives, and true negatives. So this is an example of essentially a binary prediction, and it might be running over a period of time. This one could be a sequence, a gene sequence showing the introns and exons. It could be secondary structure. It could be two different cohorts of people labeled in some way, spanning age ranges, whatever. But what we're really showing is sort of this linear graph where there's an actual observation, which is marked in blue, and then a blank line where it's either a null data or something that's different. And so these are essentially yes-no conditions, ones and zeros. So the top one in blue is the actual observation. Bottom one is the predicted one. So where the blue and the red line up, those are the true positives. Where the blank or single line and the red line up, those are false positives. That's an over-prediction, if you want. And then where the blank lines or the thin lines line up, those are the true negatives. And then where there's a blank line among the prediction, essentially an under-prediction, and then the actual observation is in blue, that's a false negative. So we can record those, we can track them, but many people, especially statisticians, especially people in the area of artificial intelligence and machine learning, like to use other terms where they combine the measure of true positives, false positives, true negatives, and false negatives. And so they use things called sensitivity and specificity. So sensitivity is essentially a measure of the percentage of false negatives. So remember, if there's an N in the word, it's percent false negatives. Specificity is the percentage of false positives. So there's a P in the word, so remember that's false positives. And then precision is a little different, but it's a measure of the percentage of positive results overall. So you can write out an equation for sensitivity and specificity. Sometimes sensitivity is called recall. And so this is just a measure of the total number of true positives divided by the true positives plus the false negatives. And the specificity is the number of true negatives divided by the sum of the total true negatives and the false positives. And then we've got precision given below, where it's just about the true positives compared to the true and false positives. So those are some equations. They're reference equations. Some people are able to memorize them. I've never been able to figure it out, but that's why we've got it on a slide. So again, there's different types of terminology that people will use. In the case of computing science, they typically report precision and recall, while in the world of biology and bioinformatics, people report specificity and sensitivity, and that's usually also consistent with a lot of biomedical conditions. Now, what you can do with a test, and depending on what you're specifically looking for, depends on essentially the cost of making errors. So if you want really high precision, or if you choose to have really high precision and high specificity values, those basically favor a conservative predictor, where there's no doubt about the correctness of the prediction. Well, if you enhance the sensitivity or recall, that's essentially favoring a more liberal predictor, where you're predicting if there's a chance of success. So in the cases of where, a prediction says, well, we'll have to chop off your arm or not, the consequences are quite serious. And so you'll change your preference or bias in terms of sensitivity and specificity. If you're trying to do a screening test, where we're just saying, let's capture most of the things, including some false positives, then you'd also bias your sensitivity and specificity in a different way. So they are related, they're connected. And the fact that sensitivity and specificity use some of the same components about true negatives and false negatives and true positives, you can actually plot those things together. And the way we plot them together is through a receiver operating characteristic curve. And I've talked about that before in our earlier lectures. But it's a graphical plot, which everyone appreciates pictures worth 1,000 words. And it's a plot of the sensitivity versus the false positive rate, technically, or as plot versus sensitivity versus specificity in some regards. So in the case of biomarkers, we often use ROC plots. And given that metabolomics and transcriptomics and others are often used in biomarker selection, makes a good choice. And biomarkers, whether it's for health and disease or whether it's for detecting differences between products or assessing different groups, they're still very useful. Now, the history for a ROC curve is that these started appearing during the Second World War, and this is how the electrical engineers were basically assessing their radar for detecting planes flying over England. And in this case, they registered things as if they were able to identify it, say it's a plane, that was a hit. If they misidentified it, it was called a miss. A false negative. A false positive was essentially a false alarm, so we get all of them set up. And then the other one is a true negative, where is a correct rejection. Now the problem was, particularly in the early days of radar, was that they would mistake planes for geese, especially with flocks of geese, which have a shape not unlike a plane, at least according to the radar. And so what they would do is they would adjust the radar setting. They had different settings, and they would track the percentage of planes that they could detect correctly. And then they would also present the percentage of geese that they got correctly, and then also the percentage of geese that they incorrectly identified. And so by tracking these numbers, then you can calculate the ROC curve. And so they would adjust their settings until they finally were comfortable. But the point is that these settings obviously produce different levels of sensitivity and specificity. And there's, obvious wins. So if the radar wasn't working or wasn't working well, they could detect the geese pretty consistently, but that wasn't very helpful. Obviously, they wanted to be able to detect more planes and less geese so they could respond properly. So ROC curves, as I pointed out before, should have a shape that looks like a log curve, so things that sort of curve up and flatten out. And I've also mentioned before that a ROC curve, which is very poor overall, would have essentially a slope of 1. It's just a straight line. And that we can measure the area under a ROC curve with a maximum area of being 1 and essentially a minimum area of being 0.5, because it's a unit or normalized measure. So what I've said before, and what I'll say again, is that generally if you can get an area under a ROC curve of greater than 0.75, that's good. It's not great, but it's often good enough to be noticed. An area under the curve of 0.5 is a random guess. And an area under the curve of 1 means that your sensitivity and specificity are perfect. So black line that goes straight up, straight across, has an AUC of 1. What looks like a purple line probably has an AUC, it looks like, of about 0.9 or 0.95. Then you can see the blue line, which is basically tracking or trending along a straight line, has an AUC of about 0.5. So that's basically a random guess. So the different colored lines are giving you essentially real data from sensitivity specificity test, and it's plotting it out. And obviously you want to see things looking more like an upside down L. So that's an introduction to ROC curves. We'll talk about these a little bit more when we explore MetaboAnalyst, but it is a tool for assessing yes-no predictions. That's different than value predictions where we use correlation coefficients. So be aware of the differences and be aware of the two different ways that we generally assess performance with the variables and their values. Now, another thing that we often have to do in metabolomics and in statistics is detect clusters of things. This is, again, where we're plotting out variables, and in some cases we're looking to see if there are trends. And it might be a case where we're initially trying to plot out a correlation in some way. And here's an example. We're trying to plot out the correlation between height and weight. And for most of you can kind of see that there's a trend in this case, and that you can probably run this through a Pearson correlation calculation and come up with a correlation of about 0.73, which is okay. But you might also be struck by the fact that it's not linear. There are two batches there. Clearly, to your eyes, there's a cluster, and if you actually label the cluster, you might find that there's something different here. In this case, these are probably, rodents of some type, and in fact, the males are consistently larger than the females. And that even though there is a trend, the two actually have different correlation coefficients based on height or weight or whatever. So clustering in this case actually might be more informative than simply trying to plot a correlation. Or maybe it's worthwhile grouping these clusters to see if there are correlations within both the male and the female. So clustering is another process that's used in statistics. And we'll use it many times when we look at multivariate statistics and principal component analysis. So clustering is something we do naturally. It's where we put objects that are logically similar in groups. So clustering is something that you'll do when you have socks from the laundry and you have to pair them for different colors or different size. And you have chosen some kind of pre-chosen standard to say, I will pair these two socks together. Some people will say, if they're only approximately the same color, I will pair them. Others will use, if they're identical in color, I will pair them. And if they're not, I will throw them out. Again, there's sort of different rules that you'll use when you have a clustering process. Now, it's different. Essentially, clustering is different than classification. In classification, you generally know or have predefined the classes. You're telling the algorithm to some extent what is supposed to happen. It has prior information. In clustering, you aren't predefining the clusters. You aren't giving a whole lot of extra information. In that regard, you're just letting sort of things fall where they may. And this is an example in terms of this height versus weight. We were just simply measuring it. That's what we observed. We didn't say, let's put all the males together and group them. And that's why, in fact, initially the data looked like this. We didn't color them. So clustering is typically a thing you do before you start with classification. Clustering is a process which says, is there actually a set of classes here that's worthwhile performing? So clustering still requires a rule, which is essentially a way to measure similarity. And so we can use things like similarity matrices or similarity scoring tables or dissimilarity coefficients or similarity coefficients. Any of these things can be used. So if we've got some measure that allows us to measure similarity, then we also have to decide on a threshold to say this is similar or this is not. It's a member of the cluster or it's not. We also have to have a defined measure of the distance between clusters. And typically we have to start with a cluster seed. So typically if you're sorting laundry, you will grab into sort of this basket of colored socks and pull out one. And then you'll start looking to see if you can find something that's similar to it. So that is the seed to your clustering process. So there are different types of clustering algorithms. One is called a Kmeans algorithm. Another is called the hierarchical clustering method. So in K-means, what you're doing is just taking n objects and dividing them into a smaller number of clusters. So 100 objects, 10 clusters. In hierarchical methods, what we're trying to do is produce a set of nested or connected clusters. And we do this sort of a progressive way. Hierarchical methods are typically done when we do gene comparisons. It's a way of doing phylogenetic comparisons, trees or family relationship trees. That's an example of hierarchical clustering. Then there's another form which is called a self-organizing feature map. Typically it's called an SOM. And that essentially produces a cluster set through a process of training. So in that regard, there may not be any defined rules. It sort of creates some rules. So for K-means clustering, this is the algorithm. And so typically the K-means algorithm is still involves choosing a seed. In this case, take your first object. And then once you have defined that or identified that, you take the next object and calculate similarity to each existing, if we want to call it a center of the cluster. If that center is greater than some threshold, we'll add that object to the cluster and then recalculate the center or the average of those properties. If we can't find something that passes that threshold, then we leave that object out. And you repeat this over and over again until everything is all appropriately clustered. So if we had this original group, we randomly choose sort of the turquoise ball here. And we have a rule that the color, the lambda value of absorbance or color, has to be close to, at least in terms of wavelength, 50 nanometers, plus or minus the color of that particular centroid. So in this case, we will randomly choose something, and we choose the ball that's just below, which is a little darker turquoise. And we'll look through our rule. Is it within 50 nanometers of the wavelength of these two, or this centroid? And in fact, they are. And so since they're within 50 nanometers, we'll say we'll join. But if we were to grab, say, the orange ball, which is way up at around 600 nanometers versus the 460 nanometers for the turquoise, it wouldn't pass. In fact, many of the other balls aren't close enough. So in fact, you end up with largely 4 different clusters, 2 that are paired and 3 that are singletons. With hierarchical clustering, what you're doing is making a pairwise comparison across all objects, and you're trying to find the 2 closest objects and merging them into a cluster. Then you find the next closest one and then merge that. And then you find the next one and merge that. And essentially, you collect and ensure that everything is somehow connected. So they don't stand apart. They, in large part, have a connection. And the connection links are drawn or created to show the hierarchical or nesting structure. So you start with your initial collection of balls, still using that same rule about wavelength and absorbance, about when they start adding to them. But we first do the most similar ones, and we can see the light blue and the turquoise perhaps joining here. And we take the next one, and the dark turquoise join. Then we take, and that probably the next one would be the dark blue. And then finally, most distant of all would be the orange ball. In the course of creating that supercluster, what we're also drawing is this linkage or connection between these things so that we can actually draw a line or a connecting rod or an edge to all of these. And so as we iterate, we first get A and B, then A and B and C, and then A and B and C and D. So what's on the right is actually showing a hierarchical clustering of, in this case, a heat map. So you can see that right away there are two large clusters, red and green. So that's your first cluster. But then you can see that within these clusters there's sometimes, and this is plotting over time, where it's black and then, say, green and then black again. So those are similar. But there are others where the trend is it's first black and then green. So that's a separate cluster within the green supercluster. And then there's more complicated profiles with the green where they're sort of up and down, up and down. So those form another set of clusters. So again, this is how, both with the similarity and then through the connection bars, you're able to come up with some sort of family tree showing the linkage between these different objects. So that's hierarchical clustering. So all of this has been trying to prepare you for essentially our discussion on multivariate statistics. So talking about correlations, talking about clustering, talking about how we assess true positives and false positives and assess sensitivity and specificity. So we had learned about univariate statistics. In the case of multivariate statistics, we're dealing with multiple variables. Multi for multiple and variate for variables. If you are measuring a population, in the case of people, we might be measuring height and weight and hair color and eye color and clothing color and whatever else. Those components represent multiple variables. So if we're measuring metabolites and we're measuring 100 different metabolites, alanine, valine, glucose, creatinine, trimethylamine, those are also multivariable descriptors of our sample. And if we're doing this on 100 rats or 100 strains of plants or 100 humans, it's the same thing. When you're dealing with multivariate statistics, you have much more complicated data to deal with, and basically you have to do something called dimension reduction. So, as I said, with metabolomics experiments, we're measuring many, many things. And the example here was just that it's inherently a multivariate task. So everything you do, whether it's metabolomics, proteomics, transcriptomics, will involve multivariate statistics. So the essence of multivariate statistics is to convert multivariate data into univariate data. If you can convert the whole thing into a univariate problem, then you can apply all the things I've just taught you about p-values and t-tests and ANOVA to the data. So how do you reduce, essentially, multivariate data to semi-univariate data? The trick is something called dimension reduction. So technically when we have hundreds of variables, we have hundreds of dimensions. And if we can get things down to 1 or 2 or 3 dimensions, then that's essentially more tractable and more amenable to things like t-tests and ANOVA. So how do you do dimensional reduction? It's by principal component analysis. And what it does is it takes a whole bunch of variables and essentially looks for their correlations. So we've learned about correlations. And by looking at those correlated or co-varying groups, we can effectively reduce the whole set to different groups or clusters of uncorrelated groups. So the uncorrelated groups are the principal components. The things that are in the groups are the correlated variables. So in this way, you can potentially reduce thousands of variables to 2 or 3 key features. So in the case of NMR spectra, we're dealing with hundreds of peaks in each of the different classes of spectra. And therefore, what we're trying to do is reduce all those hundreds of peaks or hundreds of metabolites to 2 or 3 components. What we've done here is we've reduced it to just 2 components. It's plotted on the X and Y plot. And what we're able to see are 3 clusters, which, at least to your eyes, if each of these spectra looks very similar, and there might have been 30 PAPs and 30 ANITs and 30 controls, that those would fall into those 3 blue, red, and green clusters. And one of the things that's really important to remember is that PCA is only trying to reflect what is generally or should be obvious. So if you really understand your data and you've looked at it, if you were to run a PCA analysis, it should replicate what you think or is reasonably obvious to you. Very few people really look at their data, and most people simply upload it to a standard program, press the button, and wait for the answer to fall out. And I think it's really important to at least become familiar with your data to see if there are some trends that you can see. It might be hard to describe. It might be that there's so many samples that you just can't be certain. But the point is that if you can see some trends, and I think all of you guys can see how the 3 different types of spectra are quite different there, that's all that PCA is going to do, is say, yeah, they're different. If you can't see a separation after a PCA plot, then it's probably unlikely that there's any real difference. If you can't see it and you're familiar with the data, it's also unlikely that PCA will show a clear difference. But as I say, very few people actually look at their data in detail. So one way of visualizing what PCA is doing is to think of this sort of thought experiment. We're looking at a bagel or a donut. So it has a 3-dimensional shape, and it has a hole in it. So what we're doing is we're projecting information about this bagel in space using essentially flashlights. When we shine a flashlight on one wall, we will see the bagel shadow, and it looks like an O. And then if we have another flashlight and try and project what's going to be seen on the other wall, we'll see something that looks like a sausage or a wiener. So in this case, the O projection probably captures more of the variation in terms of what is a bagel. Someone shows you a flat description that says, what is this? A lot of you will be able to guess. It's a donut or a bagel. But if we saw the other projection, almost all of you would think, yeah, it's a sausage or a wiener. So one has higher information content, and that's the largest, what's called eigenvector. That's called the principal component 1. It's given a weighting. Usually it's written on the graph, 50, 60%. And the Wiener projection has less information, so that's the second principal component. It's the second largest eigenvector in this description. It tells us a little bit about depth. It tells us it's not a 2-dimensional object, that it actually has some depth. So PCA is essentially an eigenvalue decomposition problem. So mathematically, you're doing a singular value decomposition of a data covariance matrix. So you're calculating correlations across all variables, then you're doing a singular value decomposition. In mathematical terms, it's an orthogonal linear transformation, so it transforms data in one coordinate system, which might have hundreds of dimensions, into one that could have, well, an equal number, but into eigenvalues, which represent coordinates, if you want, which capture the greatest variance. So the one with the coordinate, or the eigenvector that has the greatest variance, is identified as the first principal component. The second one is the second principal component. So what you're generating with these matrices is essentially a linear equation. One which is a set of scores, which we call eigenvectors. Those are represented as T, and a bunch of P's, which we call loadings. And so you can essentially write out a score is equal to the loadings times your data. So if X is your data, P is your set of loading values, and T is your eigenvector. So the principal component, 1, in this case, T1, represents a sum, some mathematical sum with these loadings. So scores and loadings can be plotted out, and we can create what are called scores plots and loadings plots. And we'll talk about these a little later. So this is actually an example that Roy Goodacre presented some years ago when he was actually asked. This is a specific request given to him because people identified him as being a good statistician, and they sent him data on U.S. airports. And these are from all 50 states, also included Hawaii, Puerto Rico, also Alaska. And what they gave him were the latitude of the airport, the longitude of the airport, and the altitude of the airport. And they wanted to see if there was something that would, cluster in some interesting way, and whether there's some curious relationship about airports and their altitude and their positions. So does anyone have any idea what you would likely see with this kind of data set and what clusters would show up or what you would actually see? So he did this. He actually knew what he was going to see. This is his PCA plot. So if you know a little bit about geography, you'll see that this is an exact map of the United States. And then you can see something that's a little sparse up there is Alaska. And then way off is Hawaii in the lower left corner. And then Puerto Rico is sort of in the middle. Those are the first two principal components. And these are essentially the latitude and longitude. If you plotted this in the third principal component, which is in the corner, you see some relationship with altitude. But really, what this illustrates is that principal component analysis is actually a clustering process. And it's been mathematically proven that PCA is equivalent to K-means clustering, which is what I talked about before. So this is this picture of the K-means, of just grouping and grouping and grouping. And this is essentially where you're calculating these centroids, is essentially to some extent calculating these correlations. So in some respects, it's not magical. I mean, yes, the math is tough, and most of us can't do it by hand. We depend on computers. But intuitively, what it's doing is often clustering things that are and could be somewhat obvious. Now, this is something that we understand because we understand maps, and we look at them. Obviously, if we're looking at variables and like metabolite concentrations, we're not going to see a map of the United States or a map of your body unless we happen to include that information in our clustering. But this is an example of, if given the type of information, PCA will just generate, to some extent, the obvious. So once you've done this dimensional reduction, you can now have clusters that are essentially normally distributed, which is something that we want. Remember, normal distributions, Gaussian distributions mean we can do statistics. Means we have averages, means we have a variance, a standard deviation. And this is now presented in PCA space, 2 dimensions, 3 dimensions. In those sorts of things, we can now start doing things like t-tests or ANOVA tests to see if these clusters are truly different or not. So we've now reduced thousands of variables. We now have, to our eyes, 3 clusters, 3 distributions that have a mean and a standard deviation. And we can start wondering whether these things are truly different, or maybe they're all the same. But this, as I say, allows us to start doing standard ANOVA and t-tests. So when you do a PCA plot analysis, or PCA analysis in general, you are generating these equations, which will have scores and loadings. And so we can plot the scores, and we can plot the loadings. The scores plot is the one that is most often shown. And this is an example of a 3-dimensional scores plot showing the 3 principal components, PC1, PC2, and PC3 in the X, Y, Z directions. So they're colored. And again, this is to make things a little more apparent. Normally, in truth, a PCA plot shouldn't be colored because, in fact, maybe there aren't 3 clusters here. Maybe there's just 2. but the clusterings, if the statistically significant, can be detected. A loadings plot is less commonly shown, but it's also just as important. It shows how much each of the variables, in this case the metabolites, contributed to the different principal components. So as a rule, and again, you look and compare your scores plot to the loadings plot, and you basically put them on top of each other. And if the separation between the clusters in your scores plot is in the upper diagonal and the lower diagonal, then the loadings, which are in this case these metabolites, in the upper corners and the lower corners, are the ones that are contributing most to the separation. If it's a horizontal separation, so your cluster is on the right and one is on the left, then you'd look at the metabolites sort of in the center, well, on the right, and on the left as contributing most. So this is how you can identify which metabolites or groups of metabolites are driving the separations that you're seeing in your PCA scores plots. So PCA doesn't always work. Sometimes clear clusters don't show up. In some cases, it's not obvious in the first and second one, but maybe including the third one, you start seeing some separation. And that might be that it's just a matter of having a chance to look at it around or through. Sometimes a fourth one helps separate. But not always. And going to the fifth, sixth, and seventh, it starts to get ridiculous. If you can't see separations within the first 3 or 4 principal components, then it's probably time to give up. Essentially, the groups can't be distinguished. So if a PCA can't give you a modest separation that your eyes can't detect, it's not really worthwhile trying to use other statistical methods to separate them. Now, when you think you're seeing a separation, and maybe it's not as obvious as you'd like, you can start using a technique called PLSDA, partial least squares discriminant analysis. So this is not a clustering method, it's a classification method, and you need to be aware that they are different. PLSDA is supervised, meaning that you are actually telling it that these are the clusters. Here's the answer to the test. Now do the test. PCA is unsupervised. It says, here's the test. I'll come back in an hour. What's the answer? So in some respects, when PLSDA actually has labeled data, it has an advantage. It has prior knowledge. PCA doesn't have prior knowledge. What PLSDA does with that prior knowledge is it actually can enhance the separation between the PCA groups. And it does this by rotating the PCA components in space, in multidimensional space, so you can get that maximum separation. So a classification where you've essentially told something the answers, it now becomes a predictor. It's a prediction. If you've given that prior information to your predictor, as with any predictor, and people will tell you in machine learning and anything else, you need to check to see if it hasn't been overtrained, if you haven't given it too much information, that it isn't overfitting. So this is a statistical method for assessing whether you've given it too much information and whether the separation that now looks spectacular with the PLSDA, or what you could not see with the PCA, is really real. So there are 2 ways of doing it. One is to use an R-squared or Q-squared assessment, and the other approach is to use something called permutation testing. So if you have a PLSDA model, you can quantify or assess its properness or value by getting values from that called R-squared and Q-squared. Now, R-squared and Q-squared are not commonly discussed in statistics textbooks. In some respects, I think they were an invention by Simca, which many people use. There are methods now to calculate them, but it's, as I say, it's not in the standard textbooks. So R squared is formally called the correlation index. It's not a correlation coefficient. And it refers to the goodness of fit. So just like coefficient of determination, it can vary between 0 and 1. High values are good, low values are bad. Q squared is the predicted variation of the PLSDA plot, or a measure of the quality of prediction, and that also ranges between 0 and 1. And basically, R squared and Q squared are almost the same numbers. They track very closely. So generally, R squared, a little more detail, indicates how well the PLSDA model is able to mathematically reproduce the data in the data set. As a rule, if you have R squareds of 0.2 or 0.3, that's bad. If you have R squareds of 0.7 and 0.8, that's good. So think of it as a coefficient of determination, or the square of your correlation coefficient. If you're concerned about overfitting, which can and often does happen, the Q squared tells you what's going on. So you can calculate the Q squared by doing a bit of cross-validation or permutation testing. And it'll generate a few models and generate a set of numbers. And as a rule, Q squareds don't have to be quite as high. So if things are greater than 0.5, that's generally good. Q squareds of 0.9 are very rare. So as I say, R squared and Q squared are sort of an invention of SIMCA. There's a better route which is widely used and widely understood, and this is called permutation testing. So this is a little more complicated. So here, in this case, we might have a PCA plot. I've just colored things in blue. And to your eyes, maybe there's not an obvious separation. Now, if we color things in blue and red, you can see that there's a slight trend here. It's not great. Okay, so we start bringing in our heavy artillery to see if we can get some separation here. So we run our PLSDA, and now it's rotated things very nicely. And perhaps because we were only plotting in 2 dimensions, if we plotted in 3, we might have seen a better separation. So we've run the PLSDA, it's rotated things, it's modified those principal components, and now you can see a really obvious separation. Okay, is this real? Have we given it too much information? Is it overtrained, or is it just essentially an artifact? So in permutation testing, we can test this. And what we do is we now relabel everything randomly. So we have our original data set, but what was 1 is now 7, what was 2 is now 16, what was 3 is now 28, what was 4 is now something else. So that's shown in the bottom diagram, where we now have this clustering where everything has been relabeled randomly by the computer. And you can see that, at least when we start off, it's not so obvious. The red and the blue is written under the permuted data. And then we run our PLSDA on that permuted data. And it grinds away, grinds away, grinds away, and then it comes up with its best shot, which is now in the lower right corner. And at this step, it says, I can't really see a separation. Now what you're going to do is you're going to repeat this 1,000 times. It's going to calculate these PLSDAs. It's going to relabel everything. So it's now going to say 1 is now 3, and 2 is now 1, and whatever else. And it regenerates that, calculates another PLSDA, and assesses how separate they are. And then it's going to essentially plot out on the left corner how well things are separating. And so for many of them, there's no separation. For a lot of them, there'll be some partial separation. For many others, there'll be a bit more separation. And then there might be one, and it might be the one from the top corner, that outlier. It sits way out there that I've marked with an arrow. That's the one that we got. Now this is where you can use t-tests and p-values and say, is this significant? And from most of what you know and what I taught you the last time is, yeah, it is. When you see an outlier like that, this is significant. Therefore, we can assume that, in fact, that PLSDA separation up in the top right corner is valid. It wasn't overtrained. That's a real separation. Those objects, whatever they are, and for whatever reason, are separate. You can also assess PLSDA through something called a VIP plot, just like a very, very important person, but it's called a variable importance in projection plot. So these estimate the importance of each variable in the projection used in that PLSDA model. So it's equivalent to a scores plot, but in a little simpler way. So variables can have VIP scores more than 1, and generally if they are greater than 1, they're probably important. If they have scores less than 1, then they're probably not as important. So this is an example of some VIP plots generated from a PLSDA analysis. On the bottom x-axis, you can see scores ranging from probably about 0.8 to 1 to 1.2 up to 1.6. And then these are for different conditions, different analyses, but you can see that there's certain compounds, so carnosine and carnitine and asparagine and taurine, all have values that are greater than 1.4, 1.6, or 1.8. These are significant. What's also indicated is sort of in the red and green is essentially some of them in one state are high and in another state are low, and vice versa. So in fact, you might see a couple of variables that are green-red. Then down below, you'll see, I think, C:14. It's red-green. It means it's moving in the opposite direction. So the ones that are green-red and then are related as being important, they are co-varying. But the one that's going red-green in the opposite direction is opposing that variation. And in fact, therefore, adding one in one direction and one in the other direction might in fact give you a better differentiator between the two groups. So there are other ways of doing classification, something called soft independent modeling of class analogy, orthogonal projection of least latent structures, sometimes called OPLSDA. You can use machine learning techniques called support vector machines. You can use other machine learning methods called neural nets, naive Bayes, random forest. These are all available to do classification. Some are much better than others. Some are much more sophisticated. Some can produce a result that's more understandable. Others, it's a complete mystery, a black box. It sort of depends on what you want to do. So there are different ways and different steps that you need to deal with multivariate data. The first approach when you're dealing with this wall, the data barrier, is to bring out the catapults. The catapults are typically the unsupervised methods for clustering, PCA, which is equivalent to K-means clustering. If you took psychology, there's an old technique called factor analysis, which is also a PCA equivalent. If the catapults don't quite clear the data barrier, then you can start bringing up the canons. So PLSDA is one of those canons. Linear discriminant analysis is another. OPLSDA is another one. If the canons don't knock the wall down, then you can bring out the really, really heavy artillery, and those are the machine learning techniques, which are generally more sophisticated and involve neural networks and support vector machines. So those are different things that you can use in your arsenal of tools to help get through these daunting data collections that you get from NMR or mass spec. So there's a progression. You start with PCA to see if natural clusters form, if things are separating. You have no prior knowledge. The data is unlabeled. You let things fall where they may. If things are not as obvious as you had hoped, or you think there's a trend, then you bring out the PLSDA. In this case, you're adding prior knowledge. You know the classes in advance because you've designed the experiment. And you need to see if the data can be classified. And then you need to assess whether those really do exist as different classes. So if a supervised method is used, it'll almost always generate clusters. This is where it can be misleading. So you have to assess whether those clusters are real using either R squared, Q squared, or the preferred method is labeled permutation. So supervised classification is really powerful. They learn from experience. These are machine learning algorithms in many cases. They generalize, just like humans learn. They can perform pattern recognition, just like humans do. So they can find patterns in noise, just like humans can find them. A lot of people go straight to PLSDA and they don't try any PCA. Many people don't assess the significance and don't report the R squared or the Q squared or the permutation values. And this is where a lot of mistakes have been made, and they're still made. And so if you don't assess the significance using some kind of cross-validation of your separations or your clusters, you're basically reporting artifacts. As a basic rule, if you couldn't see a separation trend from the PCA or from just looking at your data carefully before you even started the statistics, then you actually may be treading on some thin ice. As I say, it's always possible to find a separation when you deal with multivariate statistics. It's just a question of, is it real or not? So that's multivariate statistics and the end of our sort of quick lesson on statistics. And then we'll just wrap up with the usual copyright slide.